This week in lab, we are continuing our isolation techniques. Now remember why isolation is going to be so important, because you're dealing with environmental or patient samples. That means they're not pure cultures. In fact, they're going to have a high number of different microbes in them. So you're going to have to learn how to isolate out to get that specific bacteria that you want. So your simulations this week are teaching you techniques for isolating from a mixed culture and using that solid agar medium that we are using on an agar plate petri dish. So hopefully by the time this week is over, through these labs, you're going to know how to estimate the number of bacteria in a sample and how to obtain pure cultures. Now, when we talk about isolation techniques, last week you did a streak plate technique. You used a 4-quadrant streak plate. What's here is what's known as a T-streak because it's got 3 regions. But the principle is still the same in that you're going from region to region, each time diluting the amount of bacteria in order to get an isolation. This week you're going to be doing pour plate technique, and it's the same principle behind diluting the sample in order to be able to isolate. So pour plate techniques are one that not only are we trying to grow microbes, but we're also going to try to quantify them. So we're going to use that dilution technique. It uses some specialized materials for pour plates, so when you're going through your pre-lab and stuff for the simulation, make sure you're really reading about what it takes to make this process work. Now, in particular, the most crucial step of it all is that liquid agar that you're going to be using. And the most important piece of it is getting the right temperature, because you're using a liquid agar. So it started as a broth, you've added some agar, and you're trying to get it to that solid state. So it has to go through this warm temperature to get the agar to dissolve, to a cool room temperature to make it solid. Where do you use it for this process of pour plate? Well, the ideal temperature is right around 50 degrees Celsius. If you get it too much warmer than that, then it's going to be so warm that when you go to inoculate it with your bacteria, it will actually kill the bacteria because it's too hot for the bacteria. If you get it too much cooler than that, then the agar is already going to start to solidify and congeal. So when you go to pour it into your petri dish, it's not going to give you that nice filled solid plate that's going to be needed for the bacteria to grow uniformly across it. So that's a huge step. So when you're going through that simulation lab, make sure you're really paying attention to the temperature and why that's so important. Once that agar, as I said, you get the right temperature, you pour it into the petri dish. Once it gets to that room temperature, it's going to form that solid state, and bacteria are going to be able to grow wherever they have settled in that. Now, unlike what you did last week with the streak plate, where you put the bacteria on top of that solid agar and everything grew on the surface, because that bacteria is within the liquid agar, when you pour it into the petri dish, the bacteria can grow wherever it is. So it's going to be on top and within the agar. So if it's within the agar, we say it's subsurface. If it's on top, we say it's a surface colony. So be looking for that as well. Now, just a couple of highlights for you to really pay attention for when you are going through this in the simulation lab. The pour plate technique, as I said, it is based on a dilution of microbes. So you start off with your original cultures, and here it's saying it's E. coli, and then you're going to take a specified amount from the original test tube and you're going to put it into a new test tube, and then you're going to mix that, and you're going to take the same amount and you're going to stick it into another test tube, and then mix it, take another, and put it into the third, so that each time you do this, you are diluting down the sample, which is why it goes from the dark red to the light pink, so that this is known as a serial dilution. And oftentimes we do it on the factor of 10, so it's 1/10, 1/10, 1/10. That's not always, but that's a good general rule. So then once you mix, or you make your serial dilutions, then you take those serial dilutions and you pour them into the petri dish. And then as you pour them into the petri dish, again at that correct temperature, then they will solidify, you'll incubate them, and then you'll come back after the appropriate amount of time and you're going to look for that colony growth. And you can notice here the colony growth is going to vary. We don't even pour this original one because the colony growth would be so massive you wouldn't see individual colonies. But from the 3 dilutions, you can see you have a few all the way to a large number. This one is so great that the colonies are running together. So when you do a pour plate technique, which one of these would you use? Okay, you're going to have to also do a lab that has talked about the quantification using a colony counter. So after you do something like this pour plate technique, then you're going to take the plates and you're going to use a colony counter, and you're going to count the number of colonies to quantify it. Now, so when you're reading through that lab and going through that simulation, again, you need to understand how the process works, but also you need to pay particular attention to how many colonies should you be looking for on your plate when you're using a colony counter. So go back to our picture here. This one is probably going to have way too many colonies because you're not going to get a good separation, so it's not going to be a great count. This one probably might have too few on it. You're not even sure if the conditions were right or did something go wrong with the dilution. So when you do a quantification of a plate with colonies, you're going to want somewhere between 30 and 300 colonies to be present. So that's going to be your second simulation lab that you're going to be doing this week. Now, again, we talked about isolation. It's so important for getting down to pure cultures with bacteria, for sampling, for looking for causative agents of disease, etc. But there is another application that I want to discuss with you, and it's talking about viral growth. And in particular, we're going to talk about bacteriophages. Now, let me preface this and talk about viruses are chapter 5 for us in lecture. So lab and lecture are a little off just here. So I'm not expecting you to know a lot about the virus itself or its life cycle or any of that for lab. What I'm doing that piece on is just so you have some understanding of how it applies to this isolation, pour plate technique, etc. So this is actually a picture, a rendition of what we refer to as a bacteriophage. It is a virus, but it is a virus that infects bacteria only. So it's not going to infect animals, humans, etc. So it's called bacteriophage because phage means eat, bacteria is what it infects. So here's, just so you have a good understanding, this is a scanning electron microscope. So this is the bacteria, and all of these on it on the outside is the bacteriophage. Now, how a bacteriophage works is this head and tail features about it. So it's got, within this head region, its capsule, it's got its DNA, and the tails are what it uses to identify the bacteria for which it's specific and can infect. So it uses its tail, it attaches to the bacteria, it injects its DNA, and then through a series of processes, it literally takes over the bacteria, turns it into a manufacturing center to make more of it. So the bacteriophages just multiply inside that bacterial cell till they get so many that they literally burst open the bacterial cell, and it's called lysis or lytic stage, and then it releases all of these viruses. Now, sometimes this bacteriophage will go into a process of where it basically goes into a little bit of dormancy. So it will inject its DNA into the bacteria replicates, replicates and divides, and with each replication, each new cell is going to carry with it the bacteriophage DNA. And then through a trigger, an environmental trigger, it's going to tell those bacteria, oh wait, now it's time to produce the bacteriophage. So now they become that if you will, that factory for those bacteriophages, producing lots of new bacteriophages, and in the end, the bacteria still rupture and they're going to release that virus. Why this is important is because when we need to grow viruses in the lab, in particular bacteriophages, viruses grow inside of other cells. They're intracellular pathogens or parasites, if you will, so they require a host. So in order for us to work with viruses in a lab, we're going to grow bacteria first, and we're going to do that through pouring and creating a bacterial field in a solid agar plate like this. Then we're going to introduce the phages, and we're going to be looking and counting plates like this, and we're going to be looking for these little spots known as plaques. Those clear zones of plaques means it's actually where death has occurred. So that's why this was important, is because instead of looking for colonies of growth, we're looking for plaques of death, because then that represents when the phage is lysed or burst open and killed its host cells. So we don't have a simulation lab for this one, but I do want to walk through a little bit about how this would occur in a wet lab. So first we're going to do what's known as a bacterial lawn. That means that you're going to take that bacteria in a warm agar, okay? You're going to pour that into a plate. You're going to allow it to go to room temperature. You're going to allow it then to incubate, and then the bacteria is just going to grow through that whole agar like it does with your pour plate technique, and you're going to get a solid field of growth of bacteria. Then on that bacterial field, you're going to introduce the phages, or the viruses. And then you're going to let that virus incubate with that bacterial lawn, if you will. And ultimately, you will start seeing plaques, or those areas of death. Now, how do we actually get this to work? knowing that bacteria can replicate and grow so fast that we're trying to get this large field of bacteria, that lawn of bacteria. And how do we get our count so that they become 30 to 300? If we're looking for colonies at 30 to 300, we're looking for plaques at 30 to 300 because you're using the colony counting machine. And so that's where they are the most effective. So because there can be, remember from our process, I'm going to go back up a couple steps, how fast these phages can replicate and divide because they've taken over that bacteria and made it into its own manufacturer, just like bacteria can grow so long. We have to have a process to dilute these down as well. So how do we do it? Just like I do the pork plate, they use a serial dilution. So we start by doing that original concentration of the phage, in this case, doing a serial dilution, and here it's a 10-fold serial dilution. Then you're going to plate the phage onto that bacterial lawn, and then you're going to allow it to incubate, and then you're going to come back and you're going to look at your plates. So this is actually true plates from serial dilution. Here, when it was the first dilution, see how there's so much light, you can't even determine an individual plaque. Neither can you here. So these two plates are better. These are the last two plates of the serial dilution. This one you might have 30, which may mean it's enough. This one is also definitely going to have a little bit less than 300. So one of these two plates is going to be the one that you're going to want to use for counting. So again, that's what we're doing in lab this week. If you have any questions at all, just reach out and let me know.